Welcome to this explainer on recombinant DNA technology. Look, if you're here, you're likely preparing for graduate-level botany work, which means you already know just how profound an impact genetic engineering has on our entire field. But today, we're going to move right past the broad strokes. We are diving straight into the precise molecular mechanics that actually make these incredible breakthroughs possible. To set our graduate-level baseline, recombinant DNA technology, or rDNA, is, at its core, all about the engineered splicing of distinct DNA species. We take specific genetic material, isolate it, and literally fuse it with DNA from a completely different organism. The ultimate goal? To build and successfully express a totally novel chimerical genetic structure inside a brand new host. Here is our roadmap for today's protocol. We'll start by unpacking the molecular toolkit, structurally map out vector anatomy using PBR322, sequence the complete recombinant workflow, and wrap things up with transformation and expression.
Let's jump right into part 1, the molecular toolkit. We'll be looking at endonucleases, exonucleases, and ligases. Before we can build anything, we actually need our molecular scalpels. And when you're cutting DNA, you've really got two primary options. Exonucleases are kind of the blunt instruments. They indiscriminately chew back nucleotides right from the terminal ends. But for the precise recombinant work we're doing, our focus is entirely on type 2 restriction endonucleases. These sequence-specific enzymes scrutinize the DNA strand and cleave internally at very specific palindromic recognition sites.
What we are seeing here beautifully illustrates the crucial difference in cleavage mechanics. On one side, EcoRI produces a staggered cut, generating what we call complementary sticky ends. This is huge because it structurally primes the DNA to be easily reattached. On the flip side, an enzyme like SMA-Y cleaves straight across that double helix, leaving you with blunt ends. And that choice right there completely dictates the exact ligation protocol you'll have to use later on.
Moving along to part 2, vector anatomy. Specifically, we'll look at the structural biology of plasmids, focusing on PBR322. Okay, so you can cut the DNA, but now you need a delivery vehicle. Enter the PBR322 vector. Fun fact about this one, it was actually one of the very first widely used E. coli cloning vectors created in a lab. We essentially took what is basically selfish DNA from bacteria and hijacked it for our own uses. It's a compact, 4,361 base pair circular plasmid, and crucially, it exists entirely extrachromosomally. That means it replicates completely independently of the host's main cellular genome.
So, what makes PBR322 so incredibly effective? Well, any successful vector absolutely has to harbor 3 essential features. First, an origin of replication to actually initiate copying. Second, selectable markers, like, say, ampicillin and tetracycline resistance, so we can easily verify which cells successfully took up the vector. And third, multiple specific cloning sites, where our restriction enzymes will insert that foreign DNA.
Which brings us to part 3, the recombinant workflow, tracing the path from isolation all the way to ligation.
Now that our tools and vectors are established, we can step through the precise lab methodology. This standard operating protocol strictly adheres to 5 sequential steps. Isolate the pure gene, cut it at the recognition sites, amplify the material, ligate it to your vector, and finally insert it into the host.
Step 1 is all about purity. It demands absolutely pure genetic material. Your desired DNA has to be isolated from all the other cellular macromolecules through rigorous cell lysis, protein removal, and precipitation. In the lab, if your sample isn't perfectly pure here, especially if you leave stray proteins behind, the restriction enzymes in our very next step, they simply won't function.
Assuming you've got a pure sample, we move to step 2, restriction enzyme digestion. Here, we're using EcoRI to cut both the plasmid vector and our foreign gene of interest. By using the exact same enzyme on both pieces, we guarantee that we get those complementary sticky ends we talked about earlier. It structurally primes them for a seamless puzzle piece union.
But wait, before we join these molecules together, we almost always need a much higher yield of our target insert, so step 3 relies on PCR, the polymerase chain reaction. This brilliant technique exponentially amplifies a single cut fragment into literally millions of identical copies right there in a test tube. It just guarantees we have highly adequate material for the ligation phase.
And that brings us to step 4, ligation. Honestly, this is where the magic really happens. We drop in the enzyme DNA ligase, which acts as our molecular glue, to permanently bind the complementary sticky ends of our amplified foreign DNA directly to our cut plasmid vector. The result is a completely closed, ready-to-go recombinant DNA molecule.
Finally, let's look at Part 4, Transformation and Expression. This covers host integration and the ultimate transgenic results.
All right, we've built our chimera. But let's be real, it's pretty useless just sitting there in a test tube. Step 5 is insertion, and we achieve this through multiple mechanisms depending on the host. You might use bacterial conjugation, bacteriophage transduction, or direct transformation. In modern labs, we actually rely a lot on electroporation. That's where we use high-voltage electric pulses to literally shock the plasma membrane, forcing it to open up just long enough to admit our foreign plasmid.
Once that vector is inside the transformed host, we've effectively hijacked the organism's own cellular machinery. The host cell goes ahead and replicates the recombinant vector right alongside its own chromosomal DNA. And if the conditions are optimal, the host will then transcribe and translate that inserted gene, successfully expressing the target protein we manufactured.
Now, why does this matter so much for you as future botanists? Because mastering these exact molecular mechanics is how we achieve absolutely vital agricultural breakthroughs. This technology right here is the foundation for transferring nitrogen-fixing genes into cereals, engineering golden rice to combat nutritional deficiencies globally, and developing transgenic, insect-resistant crops like Bt cotton.
So as you gear up to step into the lab and run your own advanced protocols, a really vital question remains. How will you harness this recombinant toolkit? Maybe you'll utilize plant-specific vectors like the TI plasmid to cultivate the next major revolution in transgenic botany. The tools are literally in your hands now. Thanks for joining me on this deep dive.